Hi everyone, my name's Rowan, and today I'll be talking about a thesis project that was supported by the Coleoptera Society. So, extreme sexually dimorphic traits are captivating because they show us what biological forms are possible. And I'd argue that one of the coolest but least studied of these traits is the incredibly long antennae of male longhorned beetles. Within this group of more than 35,000 species, male antennae can be up to 5 times the length of the body and twice the length of female antennae. But despite this tremendous morphological variability, we know shockingly little about why some cerambycids have such long antennae, although there are a couple hypotheses. So my PhD thesis addresses this question through multiple projects, and I'll be telling you about one today that uses antennal morphology as a lens into function. And this project would not have been possible without my collaborators at the Polish Academy of Sciences and my co-advisors at the University of Toronto. Now, earlier I mentioned that there are some hypotheses about exaggerated male antennae. A couple of the key ones for longhorns and for other insects relate to the role antennae serve as sensory traits. Now, the antennae are covered in sensory hairs called sensilla that sense diverse cues, including long-range pheromones involved in mate finding. And so this brings us to our first hypothesis, which is essentially that larger antennae are adaptive because they have greater surface area and support more olfactory sensilla, thereby increasing pheromone sensitivity. And this holds for some taxa in which males have branched antennae and females produce pheromone plumes that males follow. But most cerambycids have filiform antennae, and increasing the length of these antennae is not an efficient way of increasing surface area. What's more is that cerambycid males often produce pheromones that attract both sexes. So why then would males experience stronger selection on pheromone sensitivity? Well, there's another possibility. Selection on antenna length could be targeting a different sensory function, like tactile recognition of potential mates. This idea is not new. In the 90s, Larry Hanks proposed that long cerambycid antennae are adaptive because they help males quickly find females via direct antennal contact. His study focused on one sexually dimorphic species in which males actively search host plants with antennae outstretched. When a male's antennae struck a female, he recognized her sex-specific cuticular hydrocarbon profile and attempted to mate. So, elongate male antennae may be key for rapid tactile-based mate recognition. However, this antennal reach hypothesis has not been broadly tested in Cerambycidae. Fortunately, though, our two candidate hypotheses make different and testable predictions about sensillum abundances and distributions. Under the sensitivity hypothesis, we predict a positive association between antenna size and sensillum abundance, while the reach hypothesis doesn't require this association. However, we might expect sensilla to be clustered near the tips of the antennae under the reach hypothesis. Testing these predictions requires a group that varies in antennal length and sexual dimorphism, making the Central Asian genus Anaplistes an excellent candidate. We focused on 8 of the 19 or so species and subspecies within this genus, and the taxa we chose ranged from having short, nearly monomorphic antennae to long, highly dimorphic antennae. Using Anaplistes as a case study, then we aim to quantify variation in antenna size and dimorphism and test hypotheses about antennal function using sensilla data. We met those aims by collecting multiple types of morphological data, primarily from museum samples. First, we measured antenna length and, as a body size proxy, electron length. And we also measured the lengths of the 9 antennal flagellomeres from scanning electron micrographs of the ventral surface of the antennae. We then used those same SEM images to count sensilla in 3 grid boxes on each flagellomere, and estimated mean sensillum density across the 3 boxes. Then, to estimate sensillum abundance across the segment's ventral surface, we multiplied mean sensillum density by our measure of segment area. Now, we wanted to use all of this morphological data to understand how sensillum abundance and antenna length vary across species. But to do so, we needed a phylogeny. So we reconstructed one for our focal taxa using a Bayesian analysis of morphological and molecular characters. In our downstream statistical models, we accounted for phylogenetic uncertainty by running all analyses on a distribution of 100 trees sampled from the posterior. And we integrated our antenna measurements, sensilla data, and phylogenies into Bayesian generalized linear mixed models of sensilla density or abundance. These allowed us to test the independent and interactive effects of flagellomere, sex, and flagellomere length when accounting for phylogenetic relationships. Now, we included flagellomere as a term in these models because we had sensilla data from 3 flagellomeres representing different regions of the antenna. F1 at the base of the flagellum, F5 in the middle, and F9 at the tip. And by looking at these 3 segments, we were able to test whether sensilla were clustered near the antenna tip as predicted by the antenna reach hypothesis. However, we couldn't use these models to say how the abundance of individual subtypes of sensilla changed in relation to antennal length. To do that, we zoomed in on the tip segment because it should be important for detecting both olfactory and contact cues. Across this segment, we distinguished 2 functional types of sensilla based on their distinctive morphologies. Mechanosensory sensilla caetica, which have grooved walls and pointed tips, and what we refer to as short olfactory sensilla, a term that encompasses basochonic and orosilic sensilla that have potential roles in detecting long-range chemical cues like pheromones. We used a similar modeling framework to test how the abundance of these sensilla on F9 varies in relation to sex and flagellomere length. So what did we find? Well, regarding antenna form, we saw a dramatic variation in antennal length and sexual dimorphism across the genus. Here we've plotted antennal lengths in males and females, with yellows indicating shorter antennae relative to body size and purples indicating longer antennae. We've also plotted sexual dimorphism in antennal length, ranging from slightly male biased in light gray to strongly male biased in black. But I think the coolest patterns of variation appear when we look at flagellomere lengths. So here we've plotted the distribution of flagellomere lengths for males and females in each of our focal taxa. Flagellomere number is on the x-axis, with 1 being the segment closest to the base and 9 being the tip segment. The plots are ordered according to male antenna length, so the species with the longest antennae are on the right. And if we look at the species with the longest antennae, we can see that the antennal tip is disproportionately elongated in males, suggesting that there might be something functionally important going on there. Conversely, in 2 other species with shorter but still strongly sexually dimorphic antennae, the male antenna is essentially a scaled-up version of the female antenna. Together, these plots suggest that 2 different patterns of antennal sexual dimorphism evolved in a clade of closely related species, which is surprising. I'm following up on this result currently in a couple other thesis projects, including one that I'll mention briefly at the end of my talk. But for the moment, we've documented some interesting patterns of variation in antennal length across anaplasties. And this variation provides the power for us to test our hypotheses about antennal function. Now, as a reminder, we're testing 2 hypotheses that make different predictions about the abundance and distribution of antennal sensilla. And the first of these predictions can be reframed to ask about the relationship between sensillum abundance and segment length within a flagellomere, while the second relates to sensillum distributions across the antenna and requires comparison among flagellomeres. So our models can tackle both questions, but before I get into those data, I want to explain the structure of the plots. Each panel here represents a different flagellomere, with F9 being the tip. Flagellomere length is on the x-axis and sensilla abundance is on the y. Within each panel, there will be 2 curves, one for females and one for males. The gray ribbons will be 95% credibility intervals, and triangles and circles will represent species means for males and for females. If we look within a panel, we will be able to see how sensillum abundance changes in relation to flagellomere length for that segment. And if we compare among panels, we can see whether sensilla are more abundant on particular segments. So here are the results. Visually, there seems to be a positive relationship between flagellomere length and sensillum abundance, at least for F5 and F9. It also looks like those segments have more sensilla than F1. But how strong are these patterns? To answer that question, we need to look at parameter estimates from our models. So here on the right, circles represent mean parameter estimates. Whiskers are credibility intervals, and if the credibility interval does not cross the dashed line, we can say that it impacts sensillum abundance. Now that's certainly true for the 5th and 9th flagellomeres, which have more sensilla than F1. And flagellomere length is also to the right of the dashed line, but just barely. So these results suggest that sensilla are clustered near the tip, and flagellum length is weakly related to sensillum abundance. But what do we see when we look at sensilla density? Are species with long antennae maintaining the same densities as species with short antennae? The short answer to that is no. We found a clear negative relationship between flagellum length and sensillum density, meaning that species with long antennae aren't taking full advantage of that increased surface area to pack in more sensilla. But these results don't tell us what's happening with individual functional subtypes of sensilla. To answer that question, we looked at the abundance of mechano and olfactory sensilla on segment 9. And here we saw a positive effect of flagellum length on sensillum abundance for mechanosensory but not olfactory sensilla. So contrary to what we might predict under the olfactory sensitivity hypothesis, we did not find strong evidence that olfactory sensillum abundance scales with antenna segment length. Instead, the antennal reach hypothesis better aligns with our data on sensillum abundances and distributions, suggesting that recognition of tactile stimuli may have been important in the evolution of elongate cerambycid antennae. So returning to our original questions, we found a surprising degree of antennal length variability within a cerambycid genus, including 2 distinct patterns of sexual dimorphism. And that suggests that extreme antenna lengths can evolve rapidly and via multiple mechanisms. As for antenna function, our sensilla data suggests that long antennae may be favored for their tactile function, consistent with prior behavioral work. As a final note, I want to quickly mention a related thesis chapter also funded by the Coleoptera Society that I'm working on in collaboration with the McKenna Lab at the University of Memphis and with several other cerambycid biologists. The aim of this study is to map the evolution of antenna size and segmentation patterns across more than 70 species in the tribe Tracheodorini, to which Anaplastes belongs. So far, we've assembled a large morphometric dataset for these species, and we're currently running phylogenetic analyses on an anchored hybrid enrichment dataset that we generated largely from museum material. I'd be happy to chat more about that project later, of course, if there are questions. Thanks so much for listening, and I'd be happy to take questions.